All right, guys, let's, so we are recording on air again. Hello, everybody. So I'm going to wait for everyone to start paying attention here. I said this last class, I'm going to say it again. In my opinion, this is sometimes the most difficult unit of the year just because there's a lot of new vocabulary and you really need to stay on top of it. If you don't, you're going to get behind and you're not going to be able to make a lot of connections. So if you have questions, do not be afraid to ask any questions about genetics. The nice thing about genetics is usually it's the unit where you guys get extremely curious about genetics. There's always questions about it, which is good. So what I want to do, I handed out this extra sheet here. So I don't care where you stapled in. I kind of do want to jump ahead quickly to that, if it's going to allow me. Oh, I got to do this. Once the whiteboard turns off, I have to recalibrate. All right. I'm going to rewind a little bit here. We did a lot with DNA. We did a lot with protein synthesis. From there, we hopped into evolution, and now we're hopping into genetics. All three of those topics, guys, they go hand in hand. It's a chain reaction. One will not happen without the other. So I want to rewind a little bit back to our DNA days. All right, we learned about DNA. We learned a few things. Well, we learned a lot about DNA, not just a few things. When we talked about DNA, we know that DNA is composed of what? What are the three main things that composes DNA? Nope, those are the enzymes during DNA replication. There's three main things. There's a phosphate, there's a sugar, which are found on the sides of the DNA. So in this area, we would have the phosphates and the sugars. What's found in the middle? What's found in this area? What are those called? Yeah, the G, T, A, and C, which are nucleotides. Good. The main thing that you could have or should have gotten out of understanding DNA is, depending on the order of these nitrogenous bases, depends on what protein is made. Remember, guys, what are three bases together called?
Oh, I know this.
Yeah. Not yet.
What's it start with?
Starts with a C. No.
Am I close?
Starts with a C, ends with an odon.
Codon.
There you go.
Oh, wow.
So, we have codons here. I'm going to wait until it's quiet to continue. We have codons here. Three codons are going to code for what? Amino acid. A string of amino acid makes a protein. So, understanding that the order of these, of the nitrogenous bases, plays a very important role in passing on the message to the cells, which in turn is going to create amino acids, which will make the proteins. Okay? What I want you to understand today is the relationship between DNA and chromosomes. Remember, chromosomes are highly coiled DNA. That's why I put this picture up here. You see that DNA outstretched as DNA starts to wrap. These are proteins called histones. As they wrap more and more and more, it ends up all the way down here as a chromosome. I am going to be using the term chromosome extremely often. There's no getting around it in genetics. All right? Last class, did a very brief recap. How many chromosomes do humans have? 46. Fantastic. Which means how many pairs? 23. Good. 22 of those pairs are considered autosomal chromosomes, which means they don't determine the gender or the sex of the person. The last pair, the 23rd pair, are called sex chromosomes. They determine the gender of the person. So if a person has two X sex chromosomes, what's their sex? Female. Good. Which means that if they have an X and a Y, they're a male. All right? So these are, generally speaking, the XX and XY. They're the 23rd pair of chromosomes, while the first 22 pairs are the autosomal chromosomes. All right? So let me back up all the way to the beginning here. Is this going to allow me to do this? Why is this freaking out? Here we go. All right. All right, here we go. So you've seen a lot of these terms before that we're going to go over today. I know in middle school you talked about these terms, but I also know between 7th grade and 10th grade, you forget a lot of information in science. All right? So genetics, generally speaking, is the study of heredity. Heredity, guys, is nothing more than the passing on of traits from down through your lineage. All right? And even we can go back as far as through your common ancestors. This deals with evolution. All right? So the passing on of traits, the helpful traits will be passed on, generally speaking. The harmful traits will be weeded out of the gene pool. Now, this man here is known as the father of modern genetics. His name is Gregor Mendel. He was the scientist, actually he was a monk in Austria that worked with pea plants, did very long experiments with pea plants in order to figure out the basics of heredity and how heredity works. So what did he do? He bred pea plants in order to understand inheritance, so how traits gets passed on from one generation to the next. So why is his work important? Well, because he discovered the basic principles. Now, looking back at his experiments in today's, with our understanding of heredity and genetics today, they were he did a very simple experiment, which we could reiterate, we can recreate in our classroom right here. It would just take too long, so we're not going to do it. All right? He literally bred different colored pea flowers, pea plants, to see what the outcomes would be. And based on the outcomes, he was able to come up with terms such as dominant, recessive, heterozygous, homozygous, and so on. We're going to talk about what those terms mean in a little bit. So I want to talk briefly about, you got to write this stuff down today. So while you guys are jotting this down here, I just want to talk very briefly about just the, anatomical structures of a flower, because that's going to help you understand what he did. Understand, guys, that flowers have both male and female reproductive parts. All right? They have, this picture's not a great picture, but the pollen in a flower. Does anyone know what type of cell the pollen is going to be? Male. It's going to be the sperm cells. All right? And then inside, if you look at a flower, I'm going to kind of draw my, oops. If we're to look at a flower, that's my side view of the flower, you'll see the pollen up here, and then generally speaking, you'll see a part in the middle like that. That's called the pistil. Down in here, guys, there will be ovules, which is the female reproductive part. So once pollen can get into here, it's going to pollinate the flower, which means these ovules will then pollinate into seeds. All right? So if you are talking about an apple or an apple blossom, when you eat the apple in the fall time, that's a result of those flowers in the springtime getting pollinated. They can either get pollinated by their own pollen, or they can get pollinated from another flower of the same species, pollen from another flower of the same species. So when that flower, this section here swells up, that's what the apple is, guys. You're literally eating an ovary. Yeah, how's that sound? Appetizing? Appetizing? So any fruit that you eat, it's the same basic reproductive process. Pollen, which is the male sperm, must enter into the stamen and down into this area to pollinate the ovules, which will then turn into seeds. The ovary will swell in order to protect the seeds, and that's what a fruit is. Okay?
That's actually really interesting. How does a banana reproduce?
How does a banana reproduce? Banana is a fruit. It's going to be the same. There'll be a banana flower. There's seeds. They're just very small. Yep. All right. Yeah. Yeah, they're little tiny black seeds. If you cut a banana in half, those little black dots are the seeds. All right, let's move on here. So why did Mendel choose peas? He chose to do pea plants for, hey guys. He chose to do pea plants for a few reasons. Because there's many varieties with examinable traits. All right, we're going to talk about the traits that he looked at. He had distinct control over how the pea plants mated. So he could make them self-pollinate, which means the pollen from that flower gets into the stamen and pistil of that flower. So self-pollinated, or he could have ensured that the pollen from another flower, same species, pollinated a different flower. So that's cross-fertilization. All right, so he could have done self-fertilization with them or cross-fertilization with them. And also, pea plants had many either-or characteristics that could be examined. So the flowers are either purple or white. The pea pods are either green or yellow. The seeds are either round or wrinkled. So it was just a real simple flower that he could use in order to get the either-or traits. Yes, Roz? That's a great question. Ate them. I don't know. What do you do with peas? You eat them. So another interesting fact about Mendel, he wanted to be a biology teacher, but he couldn't pass the teacher exams. So he had these brilliant ideas, and he had one of the most brilliant experiments of mankind in order to understand how traits get passed down, but yet he couldn't pass a teacher exam. So I think that might mean that I'm smarter than him? Oh, man. Note to self. Jack just said maybe the test got dumber. All right. So what is a trait? A trait is specific characteristics such as a seed color or pod color in a pea plant. Any trait, guys, that gets passed on. You could pick out traits just looking in the mirror. All right? In humans, hair color, skin color, nose, yeah, anything, eye color, freckles, no freckles, widow's peak, hitchhiker's thumb. Widow's peak? Jordan has one. She pointed it out last time. It's like the point where your hair comes down to a point on your forehead. Yep, I can go back. So with the pea plants that Mendel experimented with, guys, he looked at 7 different traits. So the next slide is going to deal with the traits that he looked at.
Hold on. 2 words. You know what?
Are we good with this one? All right. Here are the traits that he looked at with the pea plants. He looked at seed shape. So remember I said either he looked at because of either-or characteristics. Seed shape was either round or wrinkled. Seed color, either yellow or green. All right. Pod shape, pod color, flower position, meaning was the flower at the top of the plant or was it at the bottom? Plant height. So these were the traits he looked at. Now, here is a term that we talked briefly about last class, alleles. This is a term I know was mentioned and you'd learned about back in middle school, but let's re-talk about what alleles are. So if we know that we have 46 chromosomes. This is considered one pair of chromosomes. Where do you get your chromosomes from? Mom and dad. So let's just say this is pair number 1. You get one of these chromosomes from mom, you get one of these chromosomes from dad. On each chromosome, guys, these chromosomes, because these are the ones that go together, these are the homologous chromosomes, they have the same traits on these chromosomes. However, this is the big however, the traits do not need to be the same. A different version of a trait is known as an allele. This is where we talked about last class, the upper and lower case letters. All right? So if we're talking about pea plants, this position here, or locus or loci, which is a position on a chromosome, if this is the position that codes for seed shape. And we know that seed shape can either be round or wrinkled. Which shape is going to be the dominant trait, round or wrinkled? Round. How do we know? It's capitalized. All right? So round can either be shown as capital R, or what else? Very good. So round could be capital R, or capital R, lowercase r. Yellow. It's the dominant because it's shown with a big Y. It can either be big Y, or big Y, little y. Those are going to show the same feature. Gray. Big S, little s, big S, big S, big G, big G, big G, little g, big A, big A, big A, small a, big T, big T, small t. Now, these are going to show the same trait. All right, so what this means is this. If we know that, I'm going to use the term genotype right now. Genotype, guys, is the genetic code. It's these together. So if this was a big R and this was a little r, so let's say from this chromosome we got a big R, and this chromosome we got a small r. What that means, guys, is that one of the plants passed down that allele, the other gene passed on the recessive allele. If the genotype was big R, that means that both of the chromosomes contained dominant alleles. If we look over here, if it's little r, what that would mean is that the chromosomes that code for seed shape would had to have passed on, it'll pop back up, a little r, if that were the case. All right? So alleles are different forms of the same gene. That's what an allele is, different forms of the same gene. So they will be found on the homologous chromosomes. So any questions as far as understanding these are genotypes. The genotypes are, to make it very easy on you, it's the lettering. It's the lettering here, all right? We'll get into a little bit deeper explanation of it next slide or the slide from now. And then we have another term that we're going to talk about called a phenotype. Phenotype, I always think of a phenotype, starts with PH, and so does photo. It's the physical expression of this. So big R is going to show what? Round. The phenotype would be round. Big R, little r. What's the phenotype? Round. Little r, what's the phenotype? Wrinkled. Good. So it's the physical expression of the gene, or I would think of it as a photo. You can physically take a photo of the phenotype. All right, and you can see the phenotype. All right. Good. So let's just example of the 7 traits that he examined. We talked about seed shape, seed color, seed coat color, pod shape, pod color, flower position, and plant height. In your web quest that you started yesterday, some of you, or on Monday, you came across these terms, the P and the F1 generation. What do you think the P generation stands for? Parent generation. F1 is generally the first generation. All right. Then we'll go to F2, which would be the generation if we bred the F1 generation. All right. So here's what I was explaining on the previous slide. So genes. Genes are a location of a specific trait on a chromosome. So here we go. We have chromosomes here. Remember, chromosomes are just highly coiled DNA, which... What's that?
The first one's the dad one.
The first one is the dad one.
Remember we talked about last class which one came first, the father or the mother chromosome?
Oh, yes, I remember that, but that necessarily doesn't hold true. Oh. All right. I just did that so you understand that one comes from mom, one comes from dad. Oh. All right. Wow, I appreciate that. So alleles, like I explained, are the alternate forms of a gene with a slight difference in the base sequence. So we have individual A here. You notice that the chromosomes are the same. They have the same banding on them, the same pattern, which means they're going to have the same information on them, but different variations of that information. So if we're talking about eye color here, eye color is more complicated than I make it seem here. But if this is blue eyes, which is recessive, how would we show a recessive trait? With little. I'm going to say little b, This brown eyes is dominant. How would we show that?
Big B.
Big B. So therefore, if this is individual A's genotype, big B, small b, because look, we have blue eyes, which is small b, which is there. We have brown eyes, which is big B here. What color eyes is that person going to have? Brown. Because there's one dominant allele there, the dominant will always overshadow. This is going to be brown. Here, we have an allele for brown eyes. Well, this is shaded brown. They're nice to shade it for you. But that's dominant, so it's going to be big B. This is going to be big B. What color eyes will individual B have? Brown. Brown. Fantastic. And then we have lowercase b, because blue is recessive. What color eyes will they have? Blue. Very good. If you look down at the bottom here, this is one of those examples of terms that are very big, but do not get freaked out, guys, when you see a lot of terms in biology. If you understand the root words, you can make a very educated guess as what the term means. Heterozygous. What does hetero mean? Different. Heterozygous, therefore, means they have different alleles. Homozygous. Same. Homo means the same. Therefore, they have the same alleles. So that's homozygous dominant in this case because they both have dominant alleles. In this case, it's homozygous recessive because it's the same recessive alleles. So these are all terms that you're going to have to know. Okay? Questions with this slide here? All right, making our way through. Already went over this one here. Dominant versus recessive. Just talked about this, but let's rehash it here. So dominant in a pair of alleles, remember, which are the same spot on the homologous chromosomes. Remember, homologous chromosomes are two similar chromosomes that have the same genes on them. Okay, those are homologous chromosomes. Each chromosome will code for the same trait, but different versions of that trait. That's an allele. So dominant in a pair of alleles when one allele is stronger than the other. That is shown by big B, little b. All right, that's dominant. Or if it's big B, they're both dominant, so that dominant trait is going to show. So this allele is expressed as the one that you can see. So the dominant one will always show. Recessive, the weaker allele in a pair of alleles. So therefore, if we're doing, again, if it's heterozygous for a trait, this is the recessive allele. That is not going to show. When is the only time a recessive trait will show?
When there's two.
Yeah, when there's two recessives. All right, so little b, yeah, now the recessive trait will show. So it has to be paired with another recessive allele in order for it to physically express itself. All right? So some of the stuff starting to come back to you a little bit?
Yeah.
So some people are like, nope, don't remember this whatsoever. Terms I talked about previously too. So genotype, the genetic makeup of the individual. That's a genotype. And whatever letters you choose to use to represent the alleles, it really doesn't matter. But the genotype, if you're using big B, small b, big C, small c, the genetic code that you're going to use, or the genetic makeup, is the genotype. The phenotype is the physical expression. Again, I always say, generally speaking, you can see or take a photo of the phenotype. You can physically see it. So therefore, it could be the height, it could be hair color, it could be eye color, it could be nose shape, widow's peak, hitchhiker's thumb, just traits. It's things that you can see or that get expressed. That is the phenotype of that. Okay? Yes?
Does dominance of a trait vary from person to person?
No. That's a good question, though. Question was, guys, does dominance of a trait depend person to person? It does not depend person to person. It could depend by population, though. Okay? Yes. So, question was this. I think what Jack was trying to ask was, let's say someone has blue eyes, or a family has blue eyes, if more people in the family have blue eyes, does blue eyes mean that it's a dominant trait? Answer is no. Okay, it does not necessarily mean that. It's based on larger populations. All right. All right, more vocabulary that we talked about. Again, don't get freaked out by these big terms. Homo means the same. So therefore, you can see up here it says when both alleles in a pair are the same, so that could be big B, or little b. They're both considered homozygous. It's also considered to be, quote, purebred. All right? So you will see once we get into genetics problems, I may say, you know, purebred for a certain trait, you should then know it's homozygous, either dominant or recessive. Heterozygous, hetero meaning different. So therefore, big T, small t is also sometimes referred to as a hybrid. All right, I think this is the last slide. So let's look at this quickly here. Here are the traits. We did this on the previous page. But what is the genotype of a heterozygous axial plant? Axial, well, we don't quite know what axial means. Axial means where the flower position is going to be. So right here. So if it's heterozygous for flower position, what is the genotype going to be? Big A, small a. Heterozygous means different. Very good. Big A, small a. Second question, what is the genotype of a wrinkled short pea plant? So we are looking at seed shape, and we are looking at, where are we here? Plant height. So if it's wrinkled, what is the genotype going to be? RR, good. Lowercase r, and short, good. Lowercase t, very good. What is the genotype for a purebred green pod plant? So we're looking at, good. So if it's green though, right? We're looking for, I believe, color and, oh yeah, we're looking at the pod color here. Good. So purebred, big G, big G. What is the phenotype for a small s plant? Wrinkled. Very good. So remember, phenotype, you can physically see it. If it is small s, this is the phenotype. Wrinkled. It's the trait that you can see. And then what is the phenotype for a heterozygous big A, small a plant? Axial. Good. All right. So that was a very quick introduction. We will be spending, guys, probably the next month on this stuff. Ugh, I know. But genetics is cool. It really is. I think you're going to be very intrigued with genetics, have lots of questions. So let me explain what we're going to do for the rest of the class once I turn this microphone off.